Good morning, students. Hope you will be fine at your respective places. Last time we have started this process of transcription, and we know we have write down a simply a reaction that this is the heterocatalytic process, not the autocatalytic, but it is the heterocatalytic. This is the heterocatalytic process. Heterocatalytic process, and in which we will see that DNA, it gives rise to the mRNA. From DNA we will see the mRNA synthesized, and then this mRNA, from this mRNA we will see proteins are synthesized. DNA, that is called as transcription, and this is called as translation. Now first we have to see which one of them is the template strand or which of them is the coding strand, which we have already discussed, and then we will see a very important enzyme, that is the RNA polymerase. RNA polymerase plays an important role. Now in case of the prokaryotes, what we will see, RNA polymerase, it gives rise to all the three types of these RNA. But in case of the eukaryotes, we have got three different types of the RNA polymerase. For example, we have got RNA polymerase first, it gives rise to the rRNA. Then we have got the RNA polymerase second, this gives rise to the mRNA, and RNA polymerase that gives rise to tRNA. Again, RNA polymerase, it is made up of the 5 subunits. 5 subunits, which are called as one of them, a part jo hai, they are in the two parts. One part is called as the core part of the enzyme, and these are 2 alpha units, 1 beta dash unit, then 1 beta unit, and 1 what we call as the omega unit. Now what will they do? These enzymes, they perform various functions. For example, this alpha unit, it binds to the initiation protein, beta dash binds to the DNA template, and this beta unit, it binds to the nucleotide. And all these, what we will see, these core parts, they always go together. There are also 2 subunits. एक को हम बोलते हैं sigma unit, and this sigma unit is responsible for the initiation of the mRNA, and second one is the rho, this factor which helps us in the elongation as well as the termination of the RNA, which you have already discussed last time. Now today we will proceed further, beta. Now what we will try to discuss, let us now draw the transcription unit. क्या? Transcription unit. Transcription unit. Suppose these are the 2 strands of DNA, this and this. This is from 3' to 5', and now these are anti-parallel, which I have already discussed. This is from 3' to 5', like this. Now we always know, that the DNA or the RNA newly synthesized जो होता है, they always what we will see, grows towards the 3 end, that is 5' to 3', like this. So we will say RNA polymerase, it will always function, ये function किस तरह करता है, from 5 to 3 end. Now this means that it always starts from 5 and goes towards the 3 end. and this RNA polymerase जो हम बात कर लेंगे, that means this RNA polymerase, it will synthesize the mRNA only in this direction. Now what we study, if RNA is going to transcribed, then it will go from this side to the other side, from this side to this side. So this means, अगर हम कहेंगे, if RNA, it has to always go from 5 prime to this prime. So this means this RNA, it will move from this side, that is side, to this side. जो हमारी न्यू स्ट्रैंड जो होगी, दैट इज फ्रॉम 5 स्ट्रैंड 5 कार्बन to 5 end to the 3rd end. And now this strand, this red one, it acts as the template strand, and this strand, it acts as the coding strand. It acts as the coding strand. So the end which has the polarity from 3 to 5 strand is called as a template strand, and the strand which has the polarity from 5 to 3rd end, that is called as the coding strand. See here, now the 5 end of this strand, this coding strand, 5 end of this strand, this one, the 5 end of this coding strand is attached to a what we call as the promoter. If we draw here like this, this, what we will see, the 5th of this, what we will see, the 5th end of this strand, of this coding strand, is attached a promoter. So this structure जो हम ये बनाते हैं, this is called as promoter, promoter. Now this promoter, अगर हम बात करेंगे, is normally a sequence of nucleotides, and one such very common sequence is TA, dash, so on. This is called as data box, like this. So what we will see, a promoter is normally a sequence of nucleotides, and one such very important common sequence is TA, TAA, and so on. And this is called a data box, and this is the most common promoter site. This is the most common promoter site, and this promoter site is mainly attached as 5 end of the coding strand and 3rd end of the template strand. When something is on the 5 end, it is called as upstream. When something is on the 5 end, it is called as upstream, and when something is on the 3rd end, it is called as downstream. So if we are talking about the promoter site, we will see that the promoter is attached on the upstream of the coding strand and downstream of the template strand. If coding, what we will say, if the promoter, हमने बात की, so if we say the promoter is, so what we will say, if we are talking about the promoter site, we say that this promoter site is attached on the upstream of the coding strand and downstream of the template strand. At the opposite end, now एट द अपोजिट एंड, अब हम इस अपोजिट एंड की, इसकी अपोजिट में जाएंगे. एट द अपोजिट एंड इज अटैच्ड द टर्मिनेटर, is attached the terminator. The terminator towards this what we call as third end, that is the downstream. It is what we call as, it is the promoter side. Promoter side. Now in case of terminator, what we will see, the terminator is, जो हमारा terminator towards the third end, it is called as downstream, and towards the 5 end it is called as upstream. रखा याद? So this is like this. So this is the downstream and this is the upstream. Now see here, if this is our template strand, this 5 to this, no sorry, if this is our template strand, we know this template strand is always in the 3-3 towards the primer end, 3 prime and towards the 5 primer end. Now if we see and the template strand, we will see हमारी जो coding strand है, it is always from 5 strand to 5 and to third end. Now from this template strand, what we will see, जो हमारे पास new what we call as the mRNA is going to be synthesized, that will be always in the 5 and to 3 end. 5 and to 3 end. Now what happens here in this, if we are taking this as the transcription end, transcription unit, RNA polymerase, it comes and it will come and bind at the promoter site. It bind at the promoter site. This is the RNA polymerase. So the new nucleotides जो हमारे पास होते हैं, which is going to be formed, will start from here. New nucleotides like this. The new nucleotides which is going to be formed will start from here when RNA polymerase binds here. The what we call as the first factor जो हमारे पास आता है, which is going to associate with it, is this sigma factor. This sigma factor, it is responsible for the association of mRNA synthesis. अब initiation हो गया, start हो गया. This when the RNA synthesis takes place, this sigma unit जो है या sigma factor है, it get detached. Now comes the new nucleotides now are formed, comes the RHO factor which comes here, and this RHO factor, it gives rise to the new nucleotides like this. We know that the RHO factor जो है, it helps us in the elongation. Ultimately it reaches to this what we call as the terminator, and what we will see as sequence of these, a chain of these nucleotides, RNA nucleotides is formed. Ultimately this RHO factor, it also get detached, ये भी detach हो जाता है. So in this way a new mRNA, a new RNA nucleotide chain is formed, and the whole process जो हमारे पास में आती है, that जो इसमें आता है, what we call, that is called as the transcription. This was all about this transcription, and tomorrow we will proceed further. Until then, goodbye. God bless you. Thanks.